Okay, so I'm very happy to introduce the first speaker this morning, Hanji Wu from Columbia University and the Princeton Neuroscience Lab. And she's going to tell us about new methods for identifying latent manifold structures in neural data. Thank you.
Thank you, Matilda. So good morning, everyone. Thanks for coming to the talk. Today I'm gonna talk about this new methods for identifying latent manifold structure from neural data. This is the work that I've been working on with Professor Jonathan Pillow at Princeton University. Okay, so let's get down to the latent manifold idea. So latent basically means unobserved or hidden. So the question we want to ask is, can we find continuous low-dimensional manifold structure underlying high-dimensional neural activity? For example, high-dimensional spectrum. The high-dimensional here means we have a lot of neurons, so it's a big neural population. And low-dimensional structure means some structure that generate these high-dimensional spectrums, but the number of the dimension is way less than the number of the neurons. Okay, so the answer is yes. There are existing literatures showing that there exists such low-dimensional structure underlying many cortices in the brain. We usually refer to this kind of structure as latent temporal structure or latent temporal dynamics. Okay, so here is a motivating example to show why this is true. Suppose we have these three artificial neurons, neuron 1, neuron 2, and neuron 3, each color-coded in blue, red, and yellow. So if we just look at these three-dimensional neural recording or neural activity, it's not very obvious there exists any lower dimensional structure. You can also apply PCA to this neural activity. You can see three principal components, each capture a majority of the variance of the data. Okay, but this neural activity is actually generated from this three frame rates. Each color corresponds to the neuron, like the blue frame rate is neuron 1, the red frame rate is neuron 2, and etc. We can also plot the frame rate at each time in that three-dimensional frame rate space. The black ball is the instantaneous frame rates of the three neurons. You can see the black ball starts from neuron 1, curves around, and ends with neuron 3. So it looks like a three-dimensional space, but actually there exists a one-dimensional structure that is curved in the 3D space. And here is the one-dimensional latent variable X that we use to generate these three frame rates. You can think of this as the animal's position in a linear track. We also generate three Gaussian bump tuning curves for each neuron, and at each time t, we have the latent variable X. We input this X into the three tuning curves to generate the frame rate at this time point t. So this is actually the generating procedure for the three artificial neurons, but we actually use a one-dimensional latent. So this example just tried to convince you that for high-dimensional neural activity, if you use linear dimensionality reduction, you don't see a lower dimensional latent, but actually there exists some lower dimensional trajectory, which we will resort to some nonlinear dimensionality reduction method to solve it. Here, we build a nonlinear model with a Bayesian approach. So what is a Bayesian approach? Typically in Bayesian approach, there are two components. If we denote the latent to be X, we have a prior over the latent X, P of X. This tells you a priori assumption you have about the latent structure. We also have the probabilistic mapping, Py given X, that generate the spectrums or neural activity from the latent X. So if we know these two components, we can sample X star at this latent manifold P of X this latent x from P of x, and then we can generate the vector of Poisson's span rates using the probabilistic mapping. Formally, we can get the marginal probability, which is the distribution of the neural activity, by doing the integral between integral of py given x multiplying px over x. Okay, but what we really care about is the prior px and the probabilistic mapping p of x. Actually, in the literature, there are modular approaches to deal with these two components. For example, for the prior p of x, people can assume linear dynamic system, can assume switching linear dynamic system, which is a piecewise kind of linear model that approximates the nonlinear system. We have non-parametric low-dimensional nonlinear system, also the recurrent neural network, which is very popular recently. We can also have a Gaussian process to model the latent structure. I'm going to talk about this Gaussian process later for people who are not very familiar with that. For the probabilistic mapping, we can do linear Gaussian. A question?
So the x implies that both vectors of firing rates.
So
X is the previous so it implies what the prediction.
Oh, actually, so X is latent, hidden. It's not known. It's something we want to infer. And Y is the observation, is the spectrum. So that's the only thing we have. And usually Y is a neuron by time matrix. Each neuron has, so each time points, we have a population of neural recording. That's the Y, the only observation. And X has the same number of time points, but is a much lower dimensional vector. So that is something we want to learn or we want to infer from the observed spectrums. Okay, so, and for the probabilistic mapping, we can use linear Gaussian model. We can have nonlinear Poisson model, or we can have deep neural network, which map from the latent to the observation plus some noise, if to get the probabilistic mapping. Okay, but our work actually extend the probabilistic mapping by modeling something we call nonlinear tuning curves. So these two components are actually our contribution to the literature. I'm going to expand these two parts in more details later. But a high-level thing about nonlinear tuning curves, for people who might not be familiar, it basically, traditional or conventional tuning curve characterize the function between the input stimuli, the function of neuron, between the input stimuli and the output firing rates. So here we are doing similar kind of tuning curve modeling like that. Just one more slide to show the comparison in a table form. So these are the literatures that I've just shown, and showed some people use some components from the prior part, and some use the components from the probabilistic mapping part to combine to get different models. And we actually generate a new column by modeling these nonlinear tuning curves together with the Gaussian process. Okay, so yeah, for example, the PFLDS is a nonlinear mapping with a neural network, but a linear dynamic system. And these three models use nonlinear dynamic system, like recurrent neural network, but they use a linear Gaussian mapping function. Okay, so let's get down to the model with more details. The model we propose is called latent manifold tuning model. So as I just explained, we only have spectrum Y, and we want to understand or discover the latent process X that generate the spectrum Y. As I mentioned, the first component is the prior. Here, we assume the latent process X is a function of time t. So we can put a Gaussian process prior over X. Question?
So what kind of thing could X be in the setting? If Y is spectrates, X could be...
Usually we assume X is a latent variable, as a continuous variable.
What kind of thing should I be thinking about?
So I don't remember, were you there when I showed the motivating example? So you have 3 spectrums from 3 neurons, which is this, but you actually generate the spectrum from a continuous latent variable, and each time point I'm going to go through this actually, but
So essentially X is just the parameter for the curve on which we're
yeah. So yeah, that's right. So if you have suppose you have a manifold and X just just move along the manifold with a certain dynamics or something, and that will generate the spectrum. I can go. This is just a toy example. I can show you some real data later.
Can be, I know that it's the mathematical properties, just as like a variable, but could it be the movement of the rods?
Yeah, exactly. So later I'm going to show an example in hippocampal place cell, but place cell is one known to encode spatial locations. You can think of this spatial location in the cognitive states of the animal as the latent. Yeah, that might be an example. X now is a process, latent process or function of time t. And one thing you can do is you can put a Gaussian process prior over X. So Gaussian process prior is usually considered as a prior to generate functions. If it's still too hard to understand, you can just think of that using a Gaussian process prior, you can generate smooth functions, especially when you have a smoothing kernel. So this is just saying now we have a smooth latent process that ensures the temporal continuity of the latent. So this is a prior assumption we impose over the latent. Okay, now the probabilistic mapping. To achieve probabilistic mapping, we assume that spec rates are generated from nonlinear tuning curves F. But because we have this non-negativity constraint over F, we don't want to generate negative fan rates. So we put a Gaussian process prior over the log of F. So again, here, Gaussian process generates smooth functions. This is just saying we assume neurons fare continuously in the latent space. Yeah, question?
What is KT then?
Oh, yeah, so KT is the kernel for the prior over X. Kernel means, so you can think of kernel as the correlation function. It basically imposes that nearby X over time scale should be correlated with each other. So the stronger correlation is, the smoother the function. Yeah, so that's KT, basically tell you how correlated these functions are. And KX here is the same idea, but now the input to the kernel is the latent space. So instead of imposing temporal continuity over time, now we impose continuous in the latent space. So it's basically the same thing. We basically impose smoothness over both the temporal domain as well as the latent domain. So conventionally, if people are familiar with, say, visual cortex, like neurons in visual cortex, many neurons are orientation selective, then the X will be the orientation from 0 to pi, and the Y will be the corresponding fan rate. And usually that kind of tuning curves are smooth over the input stimuli space. So that's kind of the idea. But the only difference is here is the X is not the input, it's the latent space that we try to infer. Okay, so now given these two components, we can get the spec rates Y as the output of the tuning curve F over X. And finally, we got the Poisson spiking from the fan rate using Poisson spiking or distribution. Okay, so this is the way we build a model. Given such a generative model, what we can do is now at each time point t, we generate x of t from the prior distribution to get xt. We input xt into the tuning curves for all the neurons to generate the spec rates lambda at point t, at time point t. Finally, we got spec trains at time t. Okay. So just briefly go over these the generative procedure. But just a reminder, when we fit the model to the data, we only have spec train Y. We want to learn what is X. we want to learn what is F. These are unknown. So we need to resort to some inference method to learn these two components. Okay. So here I'm just going to briefly talk about this. This is probably too technical. I'd love to discuss this if you have interest, but the high-level idea is we can write down the marginal distribution Py, which is the distribution for spectrum, as the integral over these three components. The first component is the Poisson probability of spikes across all neurons in times. So this is just the Poisson spiking model. The second component is the tuning curve prior, P of F. This is a Gaussian process prior, right? And the third component is the prior over the latent process, which is the first part in the graph I just showed. And we marginalize over the latent X as well as the unobserved tuning curve to get this marginal distribution. But this integral is intractable. So we can't really just do the integral and learn the hyperparameters of the model by optimizing the likelihood. So we propose to use some decoupled Laplace approximation to marginalize over F first, then we will have Py given X, this just integrating all the tuning curves. And then you can do standard maximum a posteriori information to learn the latent path F X hat by maximizing the joint distribution of Py given X multiplied Px. So this is kind of a standard two-step approximate inference to solve the problem. But after doing this, we will get the optimal latent path X hat as well as the tuning curves F. So I'm going to basically show both X and F that are inferred from the spectrum only in the following slides. Okay. So before I get down to a real data example, I want to show this simulated example, which is kind of fun. The true three-dimensional manifold is this Mobius strip. It's interesting because the Z latent, the third dimension, is not really like a cylinder or something, or a ring. It's actually pretty complex. So here we generate the latent X from the manifold. So this is the latent we generate, and you can plot that in the 1D space, X1Z. Okay? Then we generate tuning curves for each neuron on the manifold as well. Each neuron has its own manifold, its own tuning function on the manifold. We generate fine rates and Poisson spiking model to get spectrums for 100 simulated neurons. So this is the spectrum we get from the whole generating procedure. But when we do the inference, we only know the spectrum. Okay, now first, let's take a look at principal component analysis over the spectrum. You can see you need way more than 3 dimensions or 3 principal components to capture the majority of the variance in the data. So the y-axis is the accumulated variance. And if you plot the first 3 PCs, you can get this recurrent latents like that, but nothing like a Mobius strip. Here, we show the inferred latent using our model compared with the true manifold. You can plot this inferred latent against the true trajectory we used to generate data, as well as a linear dynamic system model with actually a linear tuning curve. You can see the third dimension from the linear model is basically a straight line. You don't see the structure from the third dimension. So this is the benefit of using a nonlinear tuning curve. Okay, now let's go to some real application. Here I'm going to show the rat hippocampus example. So this is the data set from Lauren Frank's lab. Consists of 7 to 35 simultaneous recording neurons. Here I'm showing an example of 31 neurons with 15-minute recording. So if you do PCA on this data, you will find 10 principal components that capture a large majority of the variance in the data. But we can apply our latent manifold tuning curve, which is the LMT model here, using only 2 latent dimensions. And I can plot the variance that captured by these 2 latent dimensions, which is about 60%. But 60% of variance has to be captured by at least 4 principal components using a linear model. Here I'm showing the 2 latent dimensions. X1, X2 are basically the 2 dimensions in the latent space, doesn't have any physical meaning or anything, the yellow curve here. But actually we know that the rat, I think it's mouse, sorry. The mouse is traversing in this W-shaped maze. So we can get the 2D real-time location and plot the trajectory of the real location of the mouse, which is the black curve here. But actually, the 2D trajectory we learn doesn't have physical meaning. In order to plot the yellow curve on top of the black curve, we need to resort to some affine transformation, but it's linear. So after applying the affine transformation, you can see these 2 curves follow each other pretty well. So this provides some nice interpretation of the latent trajectory we found, which is the XY coordinate of the mouse in the real spatial location. But just to remember, we don't have this trajectory information when we fit the model. We only know the spectrums. So that's the nice interpretation of the model. Again, we also want to show the tuning curves. The first row is the true tuning curves, the empirical tuning curve, plot on the real locations. And this row is the estimated tuning curve F plot on the latent 2D locations. So here I'm visualizing them in a 1D case, but they're actually 2D locations and 2D tuning curves. So we can see the tuning curves, the estimated tuning curves also match the empirical tuning curve pretty well. So this tuning curve and the latent together shows that the model can fit the data well, and we have nice interpretation of the latent process or latent trajectory we find. Okay, but this is also something that we already know about because people know place cells can encode locations. So this is not that surprising to us. It's just something that we kind of find as a proof of concept. So the summary of this latent manifold tuning model is we propose this doubly nonlinear latent variable model for spectrum data. The doubly nonlinear come from 2 Gaussian processes. One Gaussian process define the latent, the other Gaussian process define the tuning curve. And the model is good for extracting some low-dimensional nonlinear manifolds from place cells, and we can recover some smooth tuning curves for individual neurons. Okay, so any question at this point? Is that a question? Yeah.
Affecting the model. What is the meaning that you chose for this? You usually have to give in your data.
Yeah. So that's right. But we deal with homogeneous Gaussian process, not inhomogeneous one. So we definitely need to bin the model at the beginning. But how much you bin that basically affects the fan rates function. You can still have different bins and fit different models. But here we try, I think our bin size is 100 millisecond for each time bin. So we definitely, you can apply to the raw data with the raw frequency they sample, they collect from the data. It always works. It doesn't matter how much you bin it or what's your bin size. You can also always infer that. It's just that the fan rate might change a bit. So, yeah.
Length of data, what is the...
No, we don't really have that kind of hard constraint. But it basically, if you have too short data, you don't have enough spectrum to tell you what the latent or what's the tuning curve should be. So we have to have enough data, especially place cells fare really sparsely. So we don't want to have very short data. Question?
PCA. Have you looked at any of the nonlinear PCAs that are available? For example, just including the sum of the squares gives you a nonlinear clustering.
I haven't looked at nonlinear PCA per se, but there's, as I just showed in the previous slides, there are many other competitive models using different nonlinear dynamic assumption and mapping functions as well. So nonlinear PCA is basically you have a nonlinear mapping from the latent to the observation, but your dynamic, a PCA doesn't have dynamic assumption. They're just static points. So I actually compare with different methods and show that the predictor for performance is better when using our model. I'm not showing the comparing bar graph and things here, but thanks for bringing that up. It's definitely necessary to compare with those methods to see whether the model we propose is more like fits the data better and as well as has better interpretation. But yeah, we do have that. Question?
Can you extract from your analysis a sense in which the response to a given neuron is complex? Using this question, you heard a lot of this field algorithms. Is the principal assumption that now set just have to feel of a given neuron this field is complex. Your analysis work better than
Okay. Yeah, I can answer your question. So convex means they only have like a bump at the location in the whole field, right? That's like the place cell. First, in many cases, place cells don't really fire at one location. They might fire at multiple locations. It's possible. So I think it's generally I think especially from experimentalists, they assume they actually, I'm not quite sure about other theory or modeling kinds of job with work with place cells, but I talked to some experimentalists. The simplest way they do is collect a histogram of the spectrums, and which you will see as a result, most of the cases they're unimodal. But in many cases, there are some scattered models in other places, not just one location. So here, in our model, we don't assume it's convex, unimodal, anything. Those are very strong function assumption. We just assume it's smooth. That's the only thing we assume. And whether... No assumption. Yeah, we don't. So we get the function from the data. If data tell us it's unimodal, it's a smooth, like concave or convex function. But if the data tell us there are multiple models, like modules in the space, then we'll learn that. So that's why we resort to this Gaussian process, which is a non-parametric way of modeling function. That means we don't assume it's Gaussian or it's convex or anything. It's just smooth. That's we feel like probably a better way of learn from the data instead of having too strong assumption or over the tuning curves or the latent. So
in the case of the place field, you do that analysis. What is the outcome? There are multiple locations with a single smooth curve.
Yeah, so this is probably an example. The true data here fair at this location, but it has this. If you plot the histogram, of course you can fit a say Gaussian function to it, and you might get a nice curve like this, but you might lose some information at this location. For example, this one you have some spectrums here. You also have more spectrum here. So if you fit one Gaussian, you might find this bump, but you might miss this location a bit more. So, I mean, this one is a nice unimodal tuning curve. I think 90% of the time it's reasonable to assume it's unimodal and you fit to the data, you get nice smooth curve. But we still think like, for example, this one, we find this double like bimodal shape, which is similar to this. But if you fit a Gaussian, you might get a wider place fills from the data, which we we think it's probably too strong to assume that way. So that's how we do that. Okay. Question?
About the convexity. So in that W-shaped maze, there's sort of areas that are unsampled, sort of like this.
Yeah.
Place field that actually is sort of centrically expanding.
Uh-huh.
Maybe like run out of it and run back into it.
Uh-huh.
One convex location in the XY plane, but you're only getting the W sample within the plane. So my running graph looks like it's not like two different place fields. Like this, my description making sense to you? You see what I mean?
So you're saying like there's a place field, and he goes in. It's like two place field.
Does your analysis modes, are you able to kind of exclude that that's what's happening, or you don't actually, your analysis doesn't make any opinion on that? You just map this W to some abstract two-dimensional plate space, and that's what you get.
Yeah, that's a good question. First, I take a look at the raw data they have from their neuron or whatever paper. My understanding of about place field is they don't have that large radius usually. So this rays, so if you look at the, not maze, raised maze, if you look at the raw, like the paper who collect and publish this data, the place field is just like, the radius is basically the width of the maze. I think that's also the reason they define, when they design the maze, they don't really design a huge arm or something, because they basically have some idea about how large the place field could be, and then they just design the maze to make sure that they won't have things like that happen. But even we have a large, even like there exists a situation that they have a large place field that cover two arms. It's possible using our model to fit that, because we don't assume where the location of the tuning curve should be, or but we can assume how wide it should be. So if it's really large and we assume the function is super smooth, like cover a large space, it's possible that we fit something like this. But if we don't have data here, it's going to be very hard, honestly. So that's okay. Question?
Yeah, given that this maze W shaped, what are the x-axis on those plots? Is that the x-coordinate, the y-coordinate?
Yeah, it's X and Y. So that's like in this example, basically the X1 is the XY location of the mouse. So you can see it's like up and down a lot.
Okay, I just what I mean then in the axis on the other plot, with actually curved plots.
Oh, sorry. Yeah, that's a good point. So here I'm showing that in one decay. So the way I do that is just.
So you're only looking for the arms, say.
No, the way I'm plotting this is actually I'm plotting this and plotting this. Okay. So from this point to this point is actually just concatenate together, but they don't have spatial continuous. I can plot that in 2D, but it's hard to plot it.
I didn't realize. Yeah. Okay.
Yeah, but that's a good point. Yeah, that's this example. Any question about the hippocampus so far? Okay, cool. Okay, next. So here, just a simple roadmap is, I'm just showing that how to model the temporal dynamics of underlying the high dimensional spectrum data. Next, I'm going to show a similar model applied to the calcium imaging to discover some static topography. The main difference is now we don't have temporal dynamics, but we just have static thing. So in which case PCA could be a simple way to fit, because PCA doesn't consider dynamics. Okay, so the high-level idea or the problem we try to ask is, what are the dimensions of olfactory perception? From perceptual judgment literature, as well as perceptual judgment plus chemical descriptor of odorants, people know that there actually exists some low-dimensional olfactory perceptual space. For example, if you present a set of odorants to a human, odorants can be described in very high-dimensional chemical feature space. The human will only tell you up to 7 or 8 different description from their own perceptual space. So actually, the perception of odorants is much lower dimension compared with the dimensionality of the odorant itself. So with this kind of prior knowledge, we want to solve the understanding of the perception with only neural response. But we want to find some lower dimensional latent olfactory perception space using neural response only. But all the previous work using human perception. They don't analyze neural response. Okay, so the neural population imaging data is from piriform cortex in a mouse, which is a pretty upstream area. And the data is collected from our experimental collaborators, Stan Pashkovsky and Bob Data at Harvard. So they got this calcium imaging data. They collect the neural response from mouse layer 2 and layer 3 neurons in piriform cortex using multiphoton microscopy in semi-paralyzed awake mouse. And they got the 2-layer of calcium imaging. And our goal is to analyze this calcium imaging neural response to understand the perceptual latent space. So again, here's the raw data. Each slide is a 2D calcium imaging along time. So we reformulate the raw data into this matrix form. It's a neuron by time now. Odorant is presented in a 2-second window, followed by 30-second blank delivery. And we calculate the sum of calcium fluorescence or response over a 2-second window after the presentation of the odorants. So we actually average the neural response within this window. So here is the raw neural data we analyze. It's ordered by neuron. Each row is the neural population to a specific odorant. Here, remember we average over the fluorescence over a window, so we don't have temporal assumption in the data. That's why we deal with static topography instead of temporal dynamics. Okay, so this is the raw data. Again, we can do PCA, right? So here we show the accumulated variance as we have more PCs. You can see here we actually analyzed 22 different odorant response. So you can plot 22 principal components to capture. You definitely need a lot of dimensions or principal components to capture the large, like the majority of the variance in the data, which tells you the data should be high dimensional, but we don't believe so. So our goal again is to find some low dimensional latent odor space, as well as the nonlinear tuning curve to map from the low dimensional space to the high dimensional neural population. And we want to infer this low dimensional latent order and the tuning curves from the data only. Here we propose this model similar to the previous one, the latent manifold tuning model with structural noise. That's a bit different. I'm going to explain this later. Okay, I'll go start from the latent space again. So we have the low dimensional order space. Here is a 2-dimensional, for example. Each dot here represent one odorant. So we put also some prior over the latent order location. Since we don't assume dynamics or temporal dependency, they're just static value with a standard normal prior. It doesn't have to be fancy or something, it's just normal prior. And then we also have the nonlinear tuning curves for each neuron. The tuning curves are also 2D because the latent space is 2D. So again, we put a Gaussian process prior over the tuning curve function. But here we are actually modeling calcium imaging data, which is Gaussian. So we allow some negative value in the frame rate. So that's why we put a Gaussian process over the tuning curve itself instead of a log of tuning curve as in previous example. But still here, K is smoothing. So the same assumption that we assume neuron fares smoothly in the latent space. So the way we generate the calcium imaging data is each time for each order, we have a latent location. We input this latent location to all the tuning curves for all the neurons, and then we can generate the neural population response for that orderand. We can repeat the same thing for the other orderands, and then we'll get the neural population response for all the orderands here. In order to generate the true observation, we need to add some observation noise, right? So here is the observed neural response is this mean response from the tuning curve plus some noise. So here, the sigma x multiplied epsilon I is what we call structure noise. You can put a heterogeneous noise like nothing dependent or no structure, but here we assume sigma x is order dependent variance, and epsilon I is neural correlated Gaussian noise. So these two part multiplying together, that give us this structure noise that explain the data better, because sometimes we assume the neural noise could be dependent on the orderands, right? Each time you present an orderand, not only the signal has correlation, but the noise has some correlation. That's our assumption. So here is, again, the generative model we built. I just explained how we can generate the neural response from the latent space. But again, when we solve the model, we only have the neural response, and we want to understand the tuning curve and the latent underlying this neural response. Okay, so I'm not going to talk about the inference methods, which is going to be quite similar. I'm just going to dive into the real data experiment analysis. But before talking about the latent estimation we have, I want to talk about the stimulus design, basically to understand what the orderants, the stimuli they use to trigger the neural response. So orderants can be represented in thousands of chemical features. And our collaborator map the thousands of chemical features down to a two-dimensional PC space, which is plot here. So this is the PC of the chemical representation of the orderants. So the first PC corresponds to increase in carbon chain length of molecules. The second PC separate different chemical functional groups. Here, our collaborator selects 22 different orderants spanning 4 different functional groups, which are color-coded here: esters, aldehydes, ketones, acids. So each color dot here is one orderant. So, and within each functional group, the carbon chain length of the molecules or of the orderants is incrementally varied. So these are the 22 orderants that peak to present to the animal to get neural response. And here I'm just showing this nice topography of these orderants in the chemical space. Okay, but this information is inaccessible for us when we do the modeling. First, I'm showing some 2D latent analysis of the orderants using PCA. So again, I already showed this figure, right? If you use PCA, the y-axis is the accumulated variance, then you might need, say, up to 20 or 21 PCs to capture all the variance. And if you look at the first two PC plots, it doesn't look that nice, okay? But if you can also plot the 2D, you can also fit our model with 2 latent dimensions. From the variance plot, we can see 2-dimensional LMT model, the latent manifold tuning model can capture almost 100% of variance in the data. And if you look at the 2D plot, so each color corresponds to the same functional group. You can see functional groups are well separated from each other. Within the same functional group, you kind of see this line structure shape, which resemble the chemical representation well. Also, within the functional group, the order of these odorants is the same as the order of the chemical space. So this provides some nice interpretation of our latent space again. The first dimension corresponds to the increase in carbon chain length. The second dimension separates the functional groups. So we assign the meaning of the latent one and the dimensionality of this latent space by post hocly look at this structure and mapping it to the chemical space. Okay, so this is some interesting, we think some interesting latent structural topography we found in the using only the neural response from in olfactory neurons. Okay, so what we can do with the latent space, right? We find this nice structure latent for 22 odorants. One thing we can do is try to see whether we can use the latent to explain the observed neural response well. One example is we show this example neuron with this estimated 2D tuning curve. We place the latent on top of the tuning curve, and then we can generate a neural response for this neuron across 22 odorants. Each color corresponds to each functional group. The solid bar here is the observed mean response rate, and the light bar here is the predicted mean response from the latent anti-tuning curve. So we can see the predicted value is pretty close to the true value, this showing that the latent space and the tuning curve together account for the neural response, the true neural data well. Okay, so this is a validation procedure we can do. Furthermore, we can use the model to predict the neural response for unseen test order. So the neuron hasn't seen this order, it just tries to predict the neural response. The way we do that is using something called cross-validation. Is some cross-validation method in unsupervised learning, which is called co-smoothing in our model. Okay, so the way that works is you have an example neuron, you head it out, and then you present the test order to all the other neurons to predict its latent location in the latent space. Then you put this latent location of this test order to the example head out neuron. You can predict the neural response for this order and compare it to the true mean response for this neuron to this order. You can comparing these two values using like R-square value or whatever value to evaluate the predictive performance of our model. Okay, so we can do the same thing for all the odorants, for all the neurons, and we can plot these R-square values comparing PCA versus our latent manifold tuning model. So we can see most of the R-square value for LMT is higher than PCA. This is showing that using our nonlinear model, we have better predictive performance. We can also, but actually this latent manifold tuning model with the red dot is independent noise. Independent noise means no structural assumption in the noise. But as I show in the modeling part, we can also assume structural noise in the observation distribution, which is the green dots. So you can see with the structural noise, it can get better predictive performance, which means it probably explains the data better. So, so basically the final conclusion of this slide is our proposed latent manifold tuning curve with structural noise has better predictive performance compared with PCA. Okay, so this is basically the end of this part. Is we propose an LMT model with structural noise. It can extract low-dimensional olfactory manifold from neural response alone, which resembles the chemical representation in the 2D space. We also identify some nonlinear tuning curves over the manifold that mapping from this latent to the neural response. Finally, we show we can have better predictive performance for neural responses, especially to head-out orders. This means like when we train a model, we don't know this order. This is just for prediction performance. Okay, So a revisit and future direction of the model is, I first show some model to model the temporal dynamic underlying the neural model. It's actually LMTM, not LTM, for spectrum data. We also show an application of this kind of model to analyze, to discover static topography underlying calcium imaging data. Some interesting future direction could be using such a method, we can examine the tuning curve and manifold changes during animal training or during animal learning. We can identify latent manifold shared across different animals. This is something that I've done a little bit already, but not showing the results. The last but not the least, in the hippocampal example, we showed that the model can discover something we have knowledge about, like the place cell in close spatial location. But what is more interesting is we don't know the neural codes. So such kind of model can help us to find some interesting latent structure or latent manifold underlying neural population data. And the order olfactory example is kind of an exemplar probe of doing that. But we are also interested in applying to more neural data where we don't yet know the code. We want to understand or discover such low-dimensional perceptual cognitive space that provides more insight about the brain and help us understand the brain better. So that's kind of our high-level goal of this kind of model. Okay, so finally, I want to thank my PhD advisor, Jonathan Pillow, and my co-authors and collaborators for these two projects. So that's the end of talk. Thanks for listening. Thank you. Question? Yeah.
Yes, very, very nice talk. There are many techniques where the goal is to reduce the effective dimensionality of the space. Principal components, independent components, nonlinear versions of each. And yours is also attempting to reduce the dimensionality. Can you say a bit about why yours is more effective or
Sure. Yeah. Sure. All this. Okay. There are. How to say. Okay. So for nonlinear methods that are like LOE, MDS, like manifold D. I don't remember D dimensions. Sorry, I don't remember what's the name for. Or isomap. These are well-known nonlinear methods, but not designed for understanding neural data. So, for example, t-SNE, right? People use t-SNE a lot to map them to low-dimensional space, to visualize them. But they're actually just trying to maintain the distance between any 2 points in the low-dimensional space, but that doesn't necessarily is like what neural dynamics should look like. So many people in our field study this neurodynamic model don't really rely on these kind of nonlinear method developed outside of the area to analyze neural data because like they're not designed for that. So, but there are a lot of methods like linear dynamic system, Poisson linear dynamic system, also Poisson nonlinear dynamics. So these are more like the people who work in this area to learn the low dimensional space. So we think those are more interpretable or should fit the neural data better. But except for this kind of motivation or the interpretability, if you just fit SOMAP to neural data and try to predict the performance, those are bad. Just look at the quantitative and also qualitative performance. Those are both worse than the model developed exactly, especially for neural data. Okay, so when you look at the model developed for neural data, I show the table that basically tell you state of techniques in this literature. The reason that we think our model should work better are twofold. The first fold is neural does have some nonlinear dynamics. Many models assume linear dynamic mainly for some model tractability, but that's not the case. For example, if you look at the hippocampus place cell, although the mouse might just traverse, so the place cell can encode the 2D location in the spatial domain, but what you actually learn from place cell, we believe, is not the real location, it's no cognitive state of the brain. For example, the mouse is standing here, so he should have a trajectory like this, but next point he might jump to other place because he's just thinking something like far from the current location. So this kind of weird behavior cannot really be modeled by linear dynamic system, so we assume there are some complex nonlinearity in the dynamic system. So that's the first fold with respect to the latent. The second part is the tuning curve. I think one of our model, the contribution of our model in the field is we introduced this, we really model the tuning curve, not just some arbitrary nonlinear mapping. People use neural network to do that, which is a well-known nonlinear function, can capture all this kind of nonlinear flexibility. But we think neurons really work in a smooth space, not just arbitrary nonlinear function. And that's why we care about this nonlinear tuning curve itself, first because it's interpretable, also it's more kind of biologically plausible and close to what a real neuron should do. And assuming there exists a nonlinear function, just assume smooth over the function is the second thing we think would be more like fits the data and explain neural data better. So maybe you apply our model to biology data, to protein data, it doesn't work. So that's kind of the motivation we have by understanding the neural data and fit the model to the data. So that's kind of the intuition. Yeah, question?
So we understand that there are some latent variables in space in the brain that some neurons tune into. So that concept of tuning curve is understandable when you have stimuli, external stimuli. But when you want to declare a goal, and you want to go into the higher cognitive states, find variables. What does it mean that some neurons have tuning curves for those?
So it just so that's why I'm saying our tuning curves is different from conventional tuning curves. Conventional tuning curves in the input is existing or observed stimulus. Here we think it works in this latent space. As I just mentioned, the place cell, I mean largely or 99% of the case place cell does encode the location, but people do observe some kind of jumpy location, like trajectory, by looking at the place field. So that's kind of the hypothesis we have, that it's not really, probably it's the noise, or it's really because it's thinking about some other location, not just the real location. So we also, place cells can also encode other things, not just location, maybe some head direction or head orientation kind of information. So I shouldn't say it's cognitive, but something that is high dimensional, definitely not just 2D. So, and the tuning curve we analyze map from the latent to the neural data. So if we have a 10-dimensional space we found, then the tuning curve would be a 10-dimensional tuning curve mapping from a 10-dimensional space to a scalar Fourier rate. So I'm not sure that explain or answer your question, but we think that the place cell might encode high dimensional information, and the function just tell you how to map this high dimensional information to the Fourier rate, not just the location, but also other information. Not sure that's helpful, but hopefully, yeah. Question?
Are we always reducing down to one dimension with the tuning curve, or is
it doesn't have to be one. It can be arbitrary, but definitely less than the neural population.
Okay.
Yeah.
With that split, like there's one lower dimension that in particular That's a better one.
Yeah, as I show in the second slide or in third slide, many other literature actually also find low dimensional trajectory, like in motor cortex, in visual cortex. Especially in visual cortex, it's kind of interesting that the latent space of visual V1 neurons actually is a three-dimensional torus. And this three-dimensional torus encodes the orientation of the bar as well as the phase of the oscillation of these neurons. So a 3D torus actually is enough to capture all this information, and maybe a large, and 3D space can capture maybe 70 or 30 or 80% of the variance of the data already. And the others are more like noise or maybe some other information that we are not familiar with. But yeah, it's pretty low dimensional. So many examples definitely show. People also argue that the brain is a much higher dimensional than the number of neurons, but that's a bit different from the way that our assumption or also other different previous finding. So yeah, we assume there's low dimensional thing. Question?
It's almost very nice. So if I would want to apply this method, because you have this very non-restricted setting, what kind of things should I be careful not to overfit my data?
First, you need to probably have enough data. That's usually a kind of a restricted, how to say, I think, okay, so one thing we check is just the average spike rate. We don't have a very low average spike rate, like lower than 0.5 per second or something. We want to make sure that we have enough spike rates that provide stronger enough data part to tell us what the function look like. But the benefit of this kind of model compared with neural network or recurrent neural network is we don't have that many model parameters to fit, which means we don't need that many data as a neural network based model to learn to really learn the latent. When the data isn't really that sufficient enough, the prior assumption we impose on the model could help. So I mean, other people have already worked with I know a postdoc who worked with gray cell also applied this kind of thing. I'm pretty surprised because I've worked with gray cell, but gray cell is it's probably denser than place fill, but it's less than place fill in hippocampus. So, which also is saying the data likelihood might be not that strong, but as long as your prior is good enough, it could help with the modeling. So I wouldn't say there's really a requirement you would consider that in order to use this kind of model preventing from overfitting. And this kind of Bayesian approach is a good approach preventing overfitting because you have Bayesian prior over that. So itself it is already pretty helpful compared with neural network or other non-Bayesian methods. I'd like to chat about what kind of data you have that could potentially use this model to fit offline or something. So, question? One more question.
Just maybe it was related to another question. So how are you picking in these scenarios, you can pick your low E number of each because you know the older setting has an XY plane of that. So, but in a more general setting where you don't have strong prior assumptions on dimensionality, how are you selecting the low E number?
Yeah, that's a good question. Actually, I have some backup slides. So, yeah, this is the example I showed previously is only 2D. I didn't say about how I get this. That's actually in my first paper that I got this results. But later I've done a bit more on the spectrum data here. I'm actually doing a type of cross-validation, as I just mentioned, which is called co-smoothing, to select the best latent dimensionality. We actually find that 3-dimensional is a better latent dimension to get the best data predictive performance, basically the fit the data better. And so there are some unpublished results. That's why I'm not showing this. This is the hippocampus stuff. So we try to understand why 3-dimensional is better than 2-dimensional. We plot the manifold and find some interesting structure from this data. But yeah, I want to go through the slides, but the main thing is we can do this kind of cross-validation thing to different data set to pick we can't really do automatic selection or learning.
Dimensions, then the rest of your parameters fall out from your optimization. So you just like, okay, so I'm going to run my data fitting with 3 dimensions. I'm going to run it with 4, but then from then on, the rest of your assumptions you fixed about the smoothing kernel and things, those just sort of fall out of the fitting process, the optimization.
Yeah, that's right. And also actually for the olfactory example, instead of selecting the latent dimensionality using cross-validation, I'm assuming the latent dimension to be 100. And I use a prior called auto-relevance determination prior, which is ARD for short. I put the prior over all the latent dimension, it will automatically select the number of dimensions to fit the data best. It's a kind of a sparsity prior. So the sparsity prior over all the dimensions that smash most of dimension on, and we end up with 15 latent dimension of the neural data. But there are some automatic selection tool that you can learn together with all the other hyperparameters of the model to give you the dimension, the effective dimension finally. So there are methods doing that.
Is that Grossberg's ART model?
Grossberg?
You mentioned ART.
ARD. Yeah, I'm not sure if people are familiar. This is actually an extension of Lasso method. Lasso assume you have the same shrink, like over all the dimension, but ARD assume different shrinkage over different dimensions. So this is a more flexible model prior compared with Lasso, which is Laplace prior. And people use that. Yeah. Question?
Can we go back to your summary slide?
This one or?
No, this one.
Okay, sorry. I have 2 summary slides.
My question is regarding the relationship between the top 2 parts. Does the olfactory manifold determination utilize the neural tuning codes either directly or implicitly in its construction?
So the way we assume these 2 components to generate data is our assumption. How neurons really do that, we don't know. But we think there is some low-dimensional space, and we think there is some mapping from this low-dimensional space to the neural data. So
I wasn't really thinking motivation. I was practically, when you actually do it, when the algorithms do it, when you fit this low-dimensional olfactory manifold for a neural response alone, does that involve the utilization of the neural tuning curves either directly or implicitly?
Yeah, I show here, right? So this is an example showing we found some latent space for all the neurons, and this latent space is shared across all the neurons. So, and then we pick this example neuron. We also estimate the 2D tuning curve. So this is the 2D tuning curve. The white means higher firing rate, and the darker means lower firing rate. And we use this tuning curve to generate neural response from the latent. Oh, okay. So that's, I don't know, that's been...
The motivation for my question is you are emphasizing the strength of your approach as you required dimensions for fitting the data of the model. But when I see a tuning curve with all of this structure in it, and look at it and it's the thesis, that's like 50 this way, 50 that way. So I got potentially, effectively, or philosophically, several hundred parameters in each of these tuning curves, and I have one tuning curve for the neuron. So in a sense, I have thousands, hundreds of thousands of independent parameters. So ask me to consider whether my interpretation of parameter in this sense is correct.
So first,
You were talking a principal component, for example, that has why so many independent components in the data. And yet you are with thousands of.
so we don't estimate these parameters. So that's like, we don't treat each parameter as independent. So that's how to say, we don't parameterize anything here. This all from the data. So we actually only estimate one parameter, which is the smoothness of the kernel. And this smoothness of kernel tell me how smooth this kernel, this tuning curve should be. And what the value of each tuning, what the value of this, each element in this tuning curve comes from the data. So that's different from assuming the tuning curve as a function or each value correspond to the latent space is an independent parameter. That's different. We actually have a much restricted parameter space compared with PCA.
How many parameters are in that?
One parameter.
So each neuron has one parameter.
Actually, we assume all the neurons share the same smoothness, so they only have one hyperparameter in the kernel. So the GP thing has a 0 and a kernel X, and this kernel is, we use a smooth kernel, which is an exponential of minus 2L squared x1 minus x2 squared. I'm not sure you can see this, but this is this x1, x2 are the latent. This is the latent. So if you ask, the kernel related only is the length scale L. This L tell you how smooth the kernel should be. The larger the L is, the wider the Gaussian is. The smaller the L is, the sharper the Gaussian is. This is the only thing we learn. But whether the shape or the value of each element comes from the data.
What I'm basically arguing is just semantics. Essentially, you're summarizing the data by this kernel of the map of data neuron.
Right.
This density function.
Right.
That you're then applying.
Right.
And I'm, I guess, wondering, that really feels like an awful lot of fitting of this high-dimensional space to this set of data to generate this smooth, this tuning function.
We can discuss later. I think we passed the time if people want to take a break. But I'd like to answer your question and just have more discussion. But yeah, let's discuss later. I don't want to go details. Okay. Okay. Thank you.